Cellular differentiation is really a product of cell division. So initially, when a sperm and an egg combine to form a fertilized egg or zygote, and then it further starts dividing, what's created are pluripotent stem cells, which mean they can potentially become any cell within the body. Now, as certain growth factors or environmental conditions act upon these stem cells, they will go down various differentiation pathways according to what genes are activated and which genes are repressed. So if you look, some of these pathways can cause them to develop into glandular cells, skin cells. They go down another pathway, they become nervous system cells, and so on. So the process of differentiation, meaning forming different types of cells as a result of environmental factors influencing the genes that are activated or repressed. So whether a cell is going through differentiation or whether it is just dividing to form new cells, the process of cell division is strictly controlled. Although we're not going through this, the process of going through the cell cycle and mitosis being part of that cell cycle is what controls cell division. Now, certain things can influence cell division, such as the size of a cell. If cells can't get too big, or else they won't be able to form the fundamental functions of a cell. If space is limited, cells will stop dividing so that things don't get too crowded. And as mentioned in the previous slide, certain hormones and growth factors can influence which genes are activated or repressed, either continuing the process of cell division or stopping the process of cell division. Now, the number of times a cell divides is related to their mitotic clock, and that mitotic clock are their telomeres. And telomeres are these repetitive sequences at the ends of chromosomes that every time a chromosome divides, the DNA within a chromosome divides, those telomeres get a little shorter and a little shorter. And a cell will stop dividing and go into either senescence or apoptosis when those telomeres get so short that if they were to divide again, they would start getting rid of genes. So even the number of times a cell can divide is under strict control as a result of these telomeres regulating the process. Now, when cell division gets out of control, we refer to that as a neoplasm or new growth, and the cells can result in what we know as a tumor. Now, there are two types of tumors that you should be aware of, and you get more of this in your clinical classes. Benign tumors and malignant tumors. Benign tumors being those which are growing slowly, they have a capsule surrounding them, they don't generally invade healthy tissue, but they can encroach on healthy tissue and put pressure on airways and blood vessels and harm the body that way. Now, malignant tumors or cancerous tumors are those which are completely unrecognizable from the surrounding cells. They, by definition, extend into surrounding tissue and take over the growth in that tissue. And they can spread to other parts of the body and take over the growth in those areas of the body as well. Now, previously, when we mentioned genes that become activated or repressed during cell division, those are the ones that typically go wrong, leading to abnormal cell growth. So a few of these genes are oncogenes. Oncogenes are genes that promote cell division, and in order to cause cancer or malignant tumors, these oncogenes become permanently turned on. Tumor suppressor genes are those genes that become activated when you want to stop cell division. And in cancerous cells, these tumor suppressor genes get permanently turned off or even lost from the genome. And then finally, genes for DNA repair, DNA damage repair, and apoptosis. If a cell gets so many mutations and becomes so unlike the surrounding cells, oftentimes DNA repair mechanisms can repair those mutations, or the cell will kill itself so that the cancer doesn't spread. Now, if those genes become inactivated, DNA repair or apoptosis genes, then of course you're not going to check that DNA damage. You're not going to kill yourself for the good of the rest of the tissue, and therefore the cell division in those cancer cells, or it's going to grow out of control. And all this slide is doing is redefining what oncogenes and tumor suppressor genes are. And this slide is telling us that we need to take into consideration that often these oncogenes or tumor suppressor genes can become activated or inactivated as a result of random mutations in the genome, but often it's environmental factors such as UV light from the sun or other types of radiation or certain chemicals. They can induce mutations in the gene in DNA, and if those mutations happen to fall into a oncogene or a tumor suppressor gene, then that can also lead to cancer. So environmental factors can play a big role in the development of tumors. Now when cells die, which they are supposed to do, they generally die one of two ways. The first is they undergo apoptosis, and apoptosis is known as programmed cell death. It's a genetic program built into every single cell that if something goes wrong or if the cells aren't needed anymore, they will automatically kill themselves for the benefit of the rest of the tissue. One example of this includes virally infected cells will often trigger apoptosis pathways so that the cell will undergo a death process that will prevent other cells from becoming infected. And the thing about apoptosis is the whole package is nice and neat and is able to be cleaned up easily by macrophages so that there's nothing left behind to harm other cells. Unlike the second type of death, necrosis, where cell death occurs due to injury, whether it be another cell dying and causing that injury, or physical injury from the environment, or lack of nutrients and oxygen, which will starve the cell and cause it to die. In the process of necrosis, the cell will burst open and release its contents. And when it releases its contents, some of those contents are lysosomes. And lysosomes, being digestive enzymes, will fuse with neighboring cells and start digesting those cells as well. And it'll create a wave of death in an area of tissue that is largely unregulated. And finally, cellular aging, something we've already talked about. The telomeres regulate how old cells can get. And when cells do get older, they wear out, they accumulate waste products, and then they die. Hopefully they die by apoptosis, but if not, they die by necrosis, which we just talked about in the previous slide. At the tissue level, loss of cells results in atrophy of that tissue or a decrease in size, and often with that decrease in size comes a loss of function as well.